Hello, my name is Daniel Farb, and today I'll be discussing a scientific article entitled "Collapse of Proteostasis Represents an Early Molecular Event in Caenorhabditis elegans Aging", written by Anat Ben-Zvi, Elizabeth A. Miller, and Richard I. Morimoto. The purpose of the experiment as a whole was to study how protein damage affects the aging process in cells, as well as at what point in the life cycle the aging starts to occur. C. elegans was used as the model organism. I will be focusing specifically on Figure 5. Figure 5 shows 4 sections: Figures 5a, 5b, 5c, and 5d. Each of the 4 parts of Figure 5 contains a different test of how the HSF1 and DAF16 stress response factors affect the expression or suppression of temperature-sensitive phenotypes compared to the control group. Figure 5a shows that, comparing to age 1 and the control group, turning off the HSF1 and DAF16 stress response factors using RNAi increases the number of relative temperature-sensitive phenotypes in the cell, which is detrimental to cell growth and leads to protein damage and cell aging. Green fluorescence protein was used to visualize the relative temperature-sensitive phenotypes. UNC15E1402 and DYN1KY51, both of which are temperature-sensitive mutant proteins found in the cells of C. elegans, were used as the templates to observe HSF1 and DAF16 compared to the control. Osmoregulation (OSM) percentage is a measure of the percentage of temperature-sensitive osmoregulation phenotypes in the cells of C. elegans. Figure 5b expresses osmoregulation percentage as a function of time in days. Therefore, Figure 5b shows that HSF1 and DAF16 RNAi-expressing bacteria both experienced a more rapid increase in osmoregulation percentage over time compared to the control group. Figure 5c shows the mislocalization percentage of the dynamin temperature-sensitive phenotype as a function of time in days. The overexpression of HSF1 and DAF16 had a significantly lower mislocalization percentage compared to the control group. Additionally, it took HSF1 and DAF16 approximately 9 days longer to achieve the peak mislocalization percentage compared to the control group. Figure 5d shows the osmoregulation percentage as a function of time in days using overexpression OE of HSF1 and DAF16 compared to the control group. Although each of the 3 groups took roughly the same path, HSF1 and DAF16 took approximately 3 and 6 days respectively to reach the same osmoregulation percentage compared to the control. We can conclude from the 4 sections of Figure 5 that the stress response factors HSF1 and DAF16 reduce the number of temperature-sensitive phenotypes. Therefore, greater suppression of temperature-sensitive phenotypes leads to less protein damage and decreased cell aging. The role of Figure 5 in the scientific article is that the stress response factors ultimately lead to the reduction of protein damage and also slow the process of C. elegans. This paper relates to and contributes to biology because, although this experiment was run on C. elegans, an organism that is much less complex than humans, the concepts and results from this experiment could hopefully one day lead to the suppression of aging in human cells. This could potentially cure diseases, as well as increase the overall lifespan of humans.